Once vision is damaged, it never comes back. That much seemed obvious. That's because when the retina is damaged, the nerve cells inside it never regenerate. But somewhere on Earth, there are creatures that can regain lost vision. Fish. Amazingly, in some fish, when the retina is injured, new nerve cells actually grow back. The secret lies in a special type of cell called Mueller glia. These cells normally support the photoreceptors deep in the retina, regulate neural activity, and keep the whole structure stable. But the moment damage occurs, Mueller glia abandon their usual roles and transform into new nerve cells. In other words, they help rebuild the retina itself, restoring lost vision. This regenerative ability is seen not only in fish, but also in some amphibians, like salamanders. But in mammals, including humans, this process doesn't occur at all. Humans do have Mueller glia in the retina, but when damage occurs, they only show a brief response, then stop completely. Why is that? Why can't human Mueller glia carry the process through? Could something be blocking their ability to regenerate? For years, scientists have been searching for clues to answer this question. And recently, a research team led by Professor Jinwoo Kim at KAIST finally uncovered a clue to the cause. Surprisingly, what was blocking Mueller glia from regenerating was a single protein, PROX1. PROX1 was already known as a protein that guides the formation of nerve cells during brain and eye development. But while examining the damaged retinas of mice, the team discovered an unusually high buildup of PROX1 inside the Mueller glia. At first, they assumed the Mueller glia themselves were producing PROX1 in response to the damage, but genetic analysis revealed that the PROX1 gene was barely expressed in Mueller glia at all. In other words, the protein hadn't been produced inside the Mueller glia. It had come from entirely different cells. This is an extremely rare phenomenon. Most proteins function only within the cells that produce them. In particular, proteins that regulate genes inside the cell nucleus almost never leave the cell. But the study revealed that PROX1 actually exits its host cell, travels into other cells, and suppresses gene expression inside them. While a few unusual proteins have been reported to move between cells before, this marked the first time such behavior had been observed in PROX1. The team traced the source of PROX1 and found that other neurons in the retina, especially bipolar cells, were the primary contributors. PROX1 produced by damaged neurons was transferred into Mueller glia, suppressing their ability to regenerate. To test this hypothesis, the team launched a series of experiments. First, they deleted the PROX1 gene specifically in the bipolar cells of mice, and then induced retinal injury. As a result, PROX1 no longer accumulated in the Mueller glia, and remarkably, the cells began to proliferate. Conversely, when PROX1 protein was introduced externally, the proliferation was once again suppressed. This confirmed that PROX1 is a key inhibitory factor controlling Mueller glia regeneration. Next, the team developed a specialized antibody that neutralizes PROX1 and injected it directly into the retina. The results were even more striking. Mueller glia in the damaged retina began to divide and started transitioning into retinal progenitor cells, the early stage of becoming neurons. Some of these cells even showed signs of having differentiated into actual neurons. The team extended their experiments to models of retinal disease. When the PROX1 antibody was injected into mice with retinitis pigmentosa, they observed not only cell proliferation, but also signs of actual vision restoration. Of course, it wasn't complete regeneration like we see in fish. The recovery effect lasted for several months, but gradually declined. In other words, the recovery wasn't permanent. But what's clear is this: regeneration, once thought impossible in a damaged mammalian retina, actually happened, and it was triggered by suppressing just a single protein. This isn't just a story about treating the retina. For a long time, we believed that once the nervous system is damaged, that's the end, especially for central nervous system cells like photoreceptors, brain cells, and spinal cord neurons. But maybe that belief was an illusion, one created by a single protein. Nature may have left the potential for regeneration within us. We just haven't figured out how to open that door yet. And once again, science is knocking on that door. And perhaps it all begins by removing just one protein,